In this video, I'm going to show how to draw the Malpighian corpuscle and label its parts. So we begin by drawing a cup-shaped structure lined by flat cells. These flat cells are the squamous epithelial cells that surround the cup-shaped structure, that is the Bowman's capsule. As a Bowman's capsule extends to form the proximal convoluted tubule, the cells there are cuboidal and show brush border epithelium. Now, the inner side of the Bowman's capsule, the squamous epithelial cells show presence of processes, so these cells are called as podocytes. Now let us draw the blood capillaries. The renal artery that enters the kidney branches to form arterioles, and these enter inside the Bowman's capsule. Once it enters inside, then it branches to form a network of capillaries, and then it moves out in the form of efferent arteriole. So the arteriole that enters inside the cup-shaped structure is the efferent arteriole, and the one that leaves is the efferent arteriole. There is a difference in the diameter between these two. The efferent arteriole is thicker or wider than the efferent arteriole. The network or the tuft of capillaries that is formed inside is called as the glomerulus. The entire cup-shaped structure here is the Bowman's capsule. So the Bowman's capsule and the glomerulus together forms the Malpighian corpuscle. This Malpighian corpuscle is located in the renal cortex. Next, we label the proximal convoluted tubule that extends from the Bowman's capsule, and here the podocytes are labeled. Podocytes help in the process of ultrafiltration. After ultrafiltration, the fluid from the blood enters inside the cavity of the Bowman's capsule. So this here is a cavity of the Bowman's capsule where the glomerular filtrate is formed. The filtrate contains the fluid part of the blood other than the large proteins and the blood corpuscles. So it is the entirely fluid part that contains all the nutrients and the waste material from the blood. This then passes down the PCT for reabsorption and urine formation. The main part here is that there are certain adaptations of this for ultrafiltration. The first one is the difference in the diameter between efferent and efferent arteriole. Now this difference creates a hydrostatic pressure in the glomerulus, which makes the fluid pass into the Bowman's capsule. So that is one part. The other is the presence of podocytes. The podocytes and the capillaries that is there in the glomerulus have small slits or pores through which the filtrate then moves into the Bowman's capsule. So this is how the process of ultrafiltration is facilitated by the Malpighian corpuscle. Ultrafiltration is the first process in urine formation.